Hello everybody, I'm going to be doing another bio video. Today we're going to be talking about CRISPR and genetic engineering. And this was a project I did with a couple of my friends. So some basic stuff on DNA replication is the process through which cells copy their genetic material in order to produce another cell. Transcription is where DNA is unzipped by the enzyme, which is DNA helicase, that is the enzyme, and each side's nucleotide to match the DNA polymerase, which is another enzyme to create a 5' to 3', which is just basically saying the amount of phosphates that is occurring on each side. So one's 5', one is 3'. And a 3 to 5' strand, that is called the lagging strand. 5 to 3' is called the leading strand. And it is some of what we call semi-conservative, meaning that when you replicate DNA, you keep some of that original DNA, but you create a new one. So you have half old, half new. So it's semi-conservative. And on top of this, we have protein synthesis as well. That comes after transcription and translation, where we have protein synthesis. Transcription is where the DNA is unzipped and we start making that new RNA strand to make an mRNA strand called messenger RNA. Basically, this carries the genetic material in order to produce a certain protein. Translation is where this RNA strand will go free-floating into the ribosome and read codons, which is a set of 3 nucleotides, which each correspond to an amino acid. These amino acids then combine to make proteins. Mutations are another thing that currently happen a lot. Some think it's rare, but most of the time mutations do occur. Some just don't change anything. Let's say a codon codes for, valine, right? And let's just say that base changes from a G to an A. So now instead of like GCA, it's GCU. And they both code for valine, so nothing has changed. However, sometimes that can cause a health problem such as sickle cell anemia, where basically that changes to another protein, which causes the cells in someone's blood cells in someone's body to be in a sickle shape where they can't get as much oxygen. There are other kinds of mistakes, cystic fibrosis, Huntington's disease, and a lot of other ones as well. But there are many different causes for this, mainly carcinogens, cancer cells, and other stuff as well. So they occur when there's a mistake in DNA replication. Mutations in DNA affect protein synthesis, where they might create a new protein that the body doesn't need. They might not create a protein that the body needs, so they might not be producing a protein that the body needs, which can lead to some very, very horrible health problems. Or it might not do anything. It might change same protein. It might not harm you in any way. So sometimes it's not all bad. So CRISPR stands for Cluster Regular Interspaced Short Palindromic Repeats. Basically what that's saying is it's a cluster of genes that you see in a certain order in space that repeats one on one another. So DNA sequences between repeats that exactly match viral sequences. It is subsequently discovered that bacteria transcribe these DNA elements to RNA upon viral infection. So with this, we are able to then swap out and precisely change certain points of genes on a person. So this was found out in 2012, that they can make it that genes can be modified in order to place desired genes in places they want using the Cas9 nuclease. Further testing of this process has allowed for them to find that this process is effectively work on other organisms. We've tested on crops, we've tested it on lab animals, we've tested it not on people, because there's some ethical conditions with that, but however, we might be getting to that point soon. CRISPR has made this so much easier because we've been able to precisely input genes at certain points now, as we discover more about the human genome, or all of the genes that make up a human. So CRISPR has really changed cancer research. So cancer is the uncontrolled abnormal splitting of cells. This causes many cells to build up in a body, and it really, really damages it, as there's an abnormal amount of cells that are not producing and eating away at the body. With CRISPR being super precise and we're learning more about the human genome, we might be able to target those certain genes that are mutated to cause cancer in tumors in order to remove that and stop it before it happens or stop it when from getting any worse. But the problem is to identify which specific gene is malfunctioning and causing the unusual division. Many treatments are available now, which involves removal of the tumor, chemotherapy, which basically injects the body with drugs that kill the cells, good and bad cells. It's very draining on the person, and to stop that division of the cells, so it helps regulate the body so that they're not constantly splitting. There's also targeting treatment where a drug is administered and targets cancer cells that is being administered as well. This is tricky though because the body won't respond in a good way because they think those cells are good, but in reality they are not. So they're going to fight back and make sure those cells are okay, but we don't want that. So, but with CRISPR though, we can edit those genes so it won't be able to replicate easily. And CRISPR has greatly affected the human genome, and due to an increase in the progression of genetic editing, we also need to know what genes we're editing, right? So all the genes in the human body, again, is called the human genome, which is composed of many different kinds of alleles and genes that make up people. This is not the only thing we need to discover as we start editing other organisms. As we discover that some certain codes start to repeat in certain places that cause the code for different things. So this certain code of protein affects your height. We could then be able to change it in a certain way eventually, so where you can get a certain desired height. You get certain desired face treatment, facial feature. You get, I don't know, longer arms, longer legs. All of these different kinds of things. But again, cancer goes into that. We can definitely go target those cancer cells really, really soon. This could lead to some great discoveries to help with sickness and disease, because we can insert genes that will be able to fight back with these viruses and bacterial infections as well. However, though, this might cause a lack in genetic diversity, because if everybody has those desired traits, everyone would be very similar. This could lead to a world where one disease is deadly to one person, and if nobody has the gene in order to fight it off, we won't be able to extract and discover that gene and give it to other people. We'll all be the same. We'll all be susceptible to disease. So, if we don't have genetic variation, though, that we will be in a run for money. Again, health and safety concerns, immorality of eugenics, concerns regarding socioeconomic status, where basically patents might be involved for this, where it will create incentive for the creation of these CRISPR and genetic findings. However, some people might try to monopolize it, which would be really, really bad. We have some funny memes at the end, so we're excited. Thank you guys.